Okay, we are good. So today, thank you everyone for coming. Today we have another really exciting seminar in our series. So thank you again that for having actually two speakers today, Carlos Lax and Andreas Guerrero. Sorry if I'm mispronouncing your name. Uh,
Andreas Andreas Guerrero.
Guerrero, thank you. And yeah, and as you all see, the title is From Host Defense to Symbiotic Partnerships in Early Diverging Fungi. So um, maybe I will, just stop here and, as the two presenters, as you guys start your talk, maybe just tell us a little bit more about yourself and then go to the presentation. Thank you.
Absolutely. Yeah. Um, oh, sorry. Yeah, my name is Andreas Guerrero. I'm a, PhD candidate and graduate research fellow at Oregon State University, where I study endosymbionts in the Mucor mycota in the Ewing lab. Carlos, if you want to say something about yourself.
Yeah, sure. I'm Carlos Lac. I just recently graduated. I'm currently a postdoc at the University of Murcia, Spain, and I study different aspects of fungal biology, focus on epigenetic regulation, but also some studies on symbiosis. That's what I'm talking to you later in the presentation. So I will hand it over to Andreas to our staff, and I will follow him with two features I think gonna be interesting for the audience.
All right, thanks. So yeah, thank you so much to the organizers for having us. I'm really excited to tell you about this really cool group of organisms and why I think that they are a great system for studying endosymbiosis. And so I'll just give you an overview of fungal symbiosis generally, and then tell you about the phylum, the Mucormycota, and then three major questions, very broad questions in symbiogenesis that we can address with this system. And then I'll finish up with just where I think the field is heading and some applications yeah, that we can use with this biology. And then Carlos will tell us about some of his ongoing work. And so, first off, um, I think we've all heard of the fungi. They've been quite trendy in recent years. Maybe you've heard of the arbuscular mycorrhizal fungi, with which 80% of terrestrial plants form crucial symbiotic relationships, or the ectomycorrhizal fungi that support forests in northern latitudes globally. Or you maybe even have heard of like the lichens. The whole concept of symbiosis was actually originally conceived to describe the lichen symbiosis between fungi and symbiotic algae and cyanobacteria. And then we're really learning a lot about how fungi and bacteria form symbioses. And I'll be telling you more about some of that work today, but just to finish out, there's also fungi that live within living plants in mutualistic symbioses, endophytic fungi, and then we find them throughout guts in mammals and reptiles and insects. Here you can see Neocalamastics, which is this basal fungus with its bizarre looking thalli, those round things, and then it has these rhizoids that penetrate cellulose and help ruminants digest cellulose. So, yeah, um, with regards to fungal bacterial interactions, there are extracellular bacteria that form important relationships with fungi, but there's also endohyphal bacteria that actually live inside of the fungal cells. And these endohyphal bacteria are found across the kingdom fungi in the dicaria, which include the Basidiomycota and the Ascomycota, and then in the early diverging fungi as well. Although I think we're calling them the zoozygomycota now. Based on spore morphology. But anyway, yeah, there's a diversity of bacteria that we find inside of the fungi. And we identify the presence of these bacteria through sequencing and then through visualization. So there are a number of methods that we can use, including scanning electron microscopy and fluorescent in situ hybridization. And that's really the gold standard for saying whether or not there is an endosymbiont in a fungus. The Mucormycota phylum is particularly well suited, in my opinion, for studying endosymbiosis. So you're probably familiar with the dikarya, and these are the Ascomycota, the yeasts, and the Basidiomycota. And they have these septate filaments, if they are filamentous fungi, that have these N plus N dikaryotic compartments. And then there's also the Mucormycota phylum, and this includes the Mucormycotina, the Mortierella and the Glomeromycotina. The Glomeromycotina are the arbuscular mycorrhizal fungi that I mentioned earlier. And then the rest of them are the environmental fungi for the most part. There's also parasites and plant-associated fungi. And there are rhizopus fungi are used in a lot of food preparations. This is tempeh here, which is made with the rhizopus fungus, but it's also a crop pathogen and a human pathogen. And these have a cellular structure of a senocytic heterokaryon, where there are not cross walls, and so the heterogeneous nuclei flow freely throughout the thallus. And one of the reasons that they are really well suited for studying endosymbiosis is the diversity of endosymbionts that we find within them and the diversity of the hosts as well. So again, there's the Glomeromycotina, the arbuscular mycorrhizal fungi, and the Mortierella mycotina, and then the Mucormycotina. And across these, we find the Burkholderia-related endosymbionts, which include Mycetohabitans and Candidaptus glomerobacter, and then the Mollicutes related endosymbionts. The MRE are mostly found in the Glomeromycotina, although some are found in Mucormycotina as well. And then the BRE are found in the Mortierella mycotina and the Mucormycotina. Um, and so this large diversity of species interactions enables a lot of comparative study and evolutionary study in how these endosymbioses have arisen. Um, and one more thing that I want to just emphasize about this particular graph is that we expect that it will be filled in a lot later. It's hard to see, but Umbellapsis we know has some endosymbionts, and so this is really preliminary, and we expect that we'll find more endosymbionts as we continue with our sampling projects as a field. Yeah, so this system is also helpful in that many of the endohypobacteria are cultivable outside of the host, and the hosts are capable of being cured of their endosymbionts. Um, and then there's a lot of interspecific variation in the host phenotype, which allows for really interesting comparative studies. And it's kind of interesting actually, and this is kind of an open question in the field. There are even within species that are hosts of endosymbionts, roughly 80% of those strains, and it varies from species to species, and sampling is not very good. But what we find is like roughly 80% of strains do not actually host bacteria and have an antagonistic response when they are exposed to endobacteria. And so this phenomenon, combined with a robust genomic resource base for the phylum, enables us to do a lot of comparative studies to understand how these interactions have evolved. They also have a really diverse and important ecology. I mentioned they are arbuscular mycorrhizal fungi, but these fungi are also just ubiquitous in the environment, and we really don't know much about like what many of them do, but we find them everywhere. And so it seems that if we can understand like endosymbiosis with these fungi, we'll understand more about just what they're doing. Finally, there are genetically tractable models available. And so Rhizopus microsporus is sort of emerging as the model system within this phylum. And Carlos, who's speaking later, actually has developed a CRISPR-based engineering platform in Rhizopus that's really doing a lot to move the field forward. And we can address a number of questions with this group of organisms. So, for example, how do endosymbioses evolve? Well, so we kind of follow the map that the insect endosymbiosis field has established for us, and we see that there's this spectrum from free living to facultative to obligate endosymbiotic bacteria, and then organelles even further down on that spectrum. And as the bacteria move through this spectrum from free living to obligate, the rate of genomic change increases and the heterogeneity of selective pressure decreases. And so this combined with a vertical transmission style and a lack of recombination within the bacteria results in a Muller's ratchet in which the genome is continually reduced in size. We see this in the endosymbionts of the fungi. And so it's hard to see, but on the left, we have a phylogeny of the Burkholdeриaceae bacteria, which make up one of the main clades of endosymbiotic bacteria in the mucormycota. And on the right of the figure, there are a number of metabolic pathways, which include cysteine biosynthesis, serine, histidine, a number of other important metabolites. And what we see is that in the facultative endosymbionts, this is mycetohabitans, um, which inhabits Rhizopus, there is some loss of metabolic pathway steps, but it's still capable of being cultivated outside of its host. In Mycoavidus, which inhabits the Mordirella mycotina, these are also facultative inhabitants. They can be cultured outside of their host, but it's very difficult. And correspondingly, they lack a lot more of the metabolic pathways. And then finally, there's the Glomerobacter, which are obligate endosymbionts, and they have very small genomes, and they cannot, at this time, be cultivated outside of the host. We also see a lot of overlap between the evolution of these endosymbionts and their hosts, and this overlap tells us that they have been interacting for a very long time and that they have co-evolved a great deal. And these relationships are believed to be roughly 350 million years old, 350 to 400 million years old, based on some genomic studies. So how are these endosymbioses maintained? So it's again, remember that roughly 80% of strains in host species don't actually harbor endosymbionts. And so in a relationship that's hundreds of millions of years old, like how is that proportion maintained? Why is it that the phenotype has not swept these populations? And then how is it that the same bacteria are able to be present in the bacteria, where in the fungi, where in insects you often see like a reduction of these endocellular bacteria to a degree where they turn into organelles and then they just disappear entirely. And yet these ones are maintained. Well, for one, these endohyphal bacteria are vertically transmitted, so that's how they're maintained within like different lines. And they exert a control over the reproduction of their hosts. So on the left, we have the sexual cycle for the mucormycota. And on the right here, I have a figure that shows what happens when host fungi are cured of their endosymbionts in two different clades of fungi. So in Rhizopus, which is a member of the Mucormycotina subphylum, the host strains are able to mate and form the sexual spore, which is the zygospore. But when cured of the endohypobacteria, they are no longer able to produce those zygospores. Uh, an inverse relationship is seen in the Mordirella mycotina subphylum, where when there are EHB present, endohypobacteria present, zygospores do not form. And the picture is not great, but when cured of their endobacteria, these zygospores, in fact, do form. We also see this dichotomy in the asexual cycle. So one of the ways that these fungi reproduce is by producing sporangiospores, which are basically clones of the previous generation. And this is Rhizopus pictured. And when it contains its endosymbiont, if it's a host strain, it produces abundant spores. However, when it is cured of its endosymbiont, it doesn't produce hardly any asexual spores. And the inverse is true in the Mordirella mycotina again, where the host strains actually really struggle to produce asexual spores. Um, and so this reveals kind of this odd dichotomy that we think is very helpful for studying how endosymbionts exert control over host reproduction. Um, and it sort of hints at what are the evolutionary pressures on both these systems that allow these endosymbionts to be maintained over such a long period of time. So in Rhizopus, it seems that the bacteria provide some benefit to their hosts. The hosts grow very well when they have their endosymbiont, and without their endosymbiont, they grow poorly. The non-hosts don't grow with the endosymbiont, but they have vigorous growth without. And that's inverted in Mortierella. Again, poor growth in the host and vigorous growth when cured. So it really seems as though the endosymbiont in the Mortierella mycotina is a parasite. And this is borne out by some research where they showed that when cured of the endosymbiont, the Mortierella mycotina, they were able to actually have a much more diverse and robust metabolism. So they produced more amino acids, more fatty acids, more organic acids when cured of their endosymbiont. And so again, this sort of prompts this question like, why are these endosymbionts maintained in the system when it seems like it's parasitic over such a long period of time, but it's not present in all, like not all of the Mortierella are necessarily vulnerable to this infection. Um, and we see a hint in Rhizopus in this sister system, where the production of beneficial secondary metabolites by the endosymbiont allows the fungus to access these new niches. So there's this important story in the field of rhizoxin, where rhizoxin is this antimitotic toxin that we believed was produced by Rhizopus microspores, and it enables Rhizopus microspores to act as a pathogen in various crop systems by killing and necrotizing plant tissue. And some work, really important work from Leila Partida Martinez showed that rhizoxin is actually produced by Mycetohabitans rhizoxinica, which is one of the endobacteria of Rhizopus. So we sort of believe that and hypothesize that secondary metabolites provide their hosts with benefits. And in the Mortierella mycotina, that there may be conditional benefits due to these secondary metabolites that allow the endosymbiosis to be maintained over a long time. And this is supported by research that shows that fungi that have the endobacteria are able to better inhibit um, phagocytic amoebae and nematodes and fungivorous protists. Um, and we see this in Rhizopus as well. And this actually relates to a related topic, which is fungal pathogenicity, where we're investigating this hypothesis that the need to evade these soil predators, these protists, has sort of prompted Rhizopus fungi to evolve ways to inhibit phagocytic cells. And this, combined with the thermotolerance of the genus, of the Rhizopus genus, has sort of pre-adapted the fungus to be really dangerous for mammals in a pathogenicity sense. And so it's actually a serious pathogen. And there's a lot of active research about how pathogenicity emerges in these fungi and what we can do to reduce that. Um, we can also address this question of how do endosymbioses begin, and sort of we have this question, open question in the field of whether or not these are antagonistic or mutualistic beginnings. So some work from Lastovetsky in 2021 showed that when non-host rhizopus were plated with the endohyphobacteria um, on a plate, they exhibited an immune reaction, an ROS response, which is shown by this blue pigment on the plate here. Whereas the host rhizopus, a cured strain plated with the endobacteria, did not produce that same ROS response. When they looked at the transcriptomics of these dynamics, they found that between the host and the non-host strains, there were very different expression profiles. However, the bacteria itself between the host and non-host strain, when co-cultured, there was not a difference on the plate here. And so that to me signals that, the fungus is sort of driving this interaction, that they are, it can kind of distinguish between a host, a beneficial associate or not. Um, Another finding that is important for considering how do these endosymbioses begin in the fungi and more broadly is this finding of sort of atypical endosymbionts in the Mucormycota. And so a strain of Rhizopus was taken from a patient in which they found Ralstonia pickettii. Ralstonia pickettii is a member of the Burkholderiaceae, but it's not a typical Burkholderiaceae related endosymbiont because it's considered free living. However, when they cured this fungus of the endosymbiont, they found that there was a reduction in the ability of the fungus to evade and inhibit um, phagocytes, including macrophages and dictyostelium. And here we have some dictyostelium that are being deformed in culture with extracts of the Ralstonian fungus co-culture. And then finally, some work that was presented in this seminar last spring from Julia Vorholt and Gabrielle Geiger showed that non-host rhizopus can be coaxed to accept some bacteria, but not others. It was prompted to, it was injected with one of the endohypobacteria, Mycetohabitans rhizoxinica, and over iterative rounds of evolution was able to form a tentative relationship. But the same did not happen when injected with E. coli. So I kind of see this field as moving towards mechanistic explanations. I think that a lot of work remains in sampling diversity to see what kinds of endosymbionts are present within the fungi, but a lot of work from Carlos and others and is sort of enabling us to understand more about mechanism. And so Carlos developed this method to use uridine auxotrophs as a selective background to do engineering in Rhizopus. And it's necessary to have these auxotrophs because these fungi are very resistant to most antifungals and have really inconsistent reactions to antifungal chemicals. And so the auxotrophy method is better. And so I've been doing this myself to try to develop a background, and I've identified a uridine auxotroph, and I'll be doing some work along these lines. The Vorholt lab and I think we have some folks from that lab in attendance today, is really pioneering this microinjection method, where they can put sort of these novel bacteria into the fungi through injection, and they presented on this in the spring. They just published a paper last week that I haven't had the opportunity to read yet, but I'm really excited. And yeah, it's just revolutionizing the field. And then, we're getting into sort of understanding how epigenetic regulation can affect these relationships as well. And Carlos will tell you more about that in a second. My work is mostly focused on trying to scale our characterization of the fungi to better understand how virulence emerges in Rhizopus. And so I'm using some dictyostelium-based assays to screen um, a large collection of Rhizopus isolates that contain endosymbionts and do not contain endosymbionts for different phenotypes. And so what we do is we collect spore exudates and media to create a spore-conditioned media. And then in one of our assays, we grow dictyostelium with the spore conditioned media and see differences in growth rates based on the conditions. And I'm still sort of getting the assay to work, but what you'll see are different um, cell counts and cell densities based on different conditioned media. And this is from the same paper where they were talking about the Ralstonia pickettii, just to give you an idea of the kind of data that this assay produces. Finally, when we're thinking about applications of this work, I think there are myriad applications, both in industry and in other fields of biology to try to understand endosymbiosis and symbiogenesis generally. So a really cool finding that didn't get a lot of attention, but I think it should get more attention is this finding from the Bonita lab, in which a Mortierella species, well, Linnamania now, was co-cultured with an a eukaryotic algae, Nanochloropsis. And with very little prompting, they were actually able to get um, the algae to be taken up by the fungus and for photosynthetic mycelium to be stabilized um, over several generations. And I just thought that was absolutely wild. It makes me think of some work that Lynn Rothschild is doing with NASA to develop these artificial lichens, and Rolando Perez too out of OpenFung and yeah, in the ND lab, I think. Anyway, they're looking at these artificial lichens as sort of being applied towards space exploration. And I really think that, I don't know, I think endosymbiosis would be a more efficient approach to a whole culture of bacteria and fungi. Sort of more down to earth, I'd be very curious to think about how we can improve efficiency of food production. Rhizopus is already used in food production. And then the mucormycota are really important producers of a lot of interesting natural products. And I'd be curious about how we can mesh bacterial metabolism and eukaryotic metabolism to produce all kinds of products that would be really difficult to do otherwise. And then finally, just keeping in mind some basic facts. From an agriculture perspective, 80% of terrestrial plants form these relationships with AM fungi, which are in the glomeromycotina, which are mucormycota, and they have their own endosymbionts that I didn't talk about that much here, but they do. And then, can we use these endosymbioses to try to better improve responses to fungal pathogens in crops and in humans? And then finally, these are really weird organisms. I didn't get to talk about it very much, but they have very bizarre genomic structures and nuclear structures, and they're cenocytic syncytia, So like there's this hypothesis that the last eukaryotic common ancestor was actually a syncytium. And I don't know if that's like a widely accepted view, but if we want to test, for example, like hypotheses around the movement of endosymbionts with nuclei and the interaction of nuclei with endosymbionts and the movement throughout the whole thallus. I think that these systems would be great for looking at some stuff like that. So just some ideas. I'm going to turn it over to Carlos so that he can talk about some of his ongoing work. But I'll be happy to take questions at the end.
Go ahead and share my screen. Hopefully you can see my presentation. And I'd like to begin thanking the organizers for the invitation and for having us here today talking about this not very conventional system. But I think that I'm bringing some very interesting stuff to the symbiosis study. And I'm going to be focusing on two very different topics, but two topics that I think that show the relevance of this kind of symbiosis, but also that show how this very close relationship between both partners are influencing each other biology. And I'm going to be focusing mostly on the Rhizopus habitat system that I studied during my PhD work. And the first topic is going to be about the control that bacteria made about sexual sporulation and about propagation of the fungus. Um, but also how this is coordinated with the environment and also how some environmental factors could replace the presence of bacteria. That's what we found. So I'm going to go back to that diagram that Andreas already showed about the life cycles of mycota because I think it's important to understand that to understand what I'm going to present next. So as he mentioned, these mycota have these spores that we call sporangiospores that they germinate and form a new mycelia. And from these mycelia, they appear these structures we call sporangia that contains these sexual spores, and they will be spread in nature and originate new individuals. But they also have this sexual cycle that happens with two mycelia from opposite mating type come into contact. And the contact happens, they form these structures that are eukaryotic that are known as zygospores. And then recombination and meiosis happens and a new individual is formed. So the Rhizopus macetoretan system has some very cool and interesting feature that Andreas presented. So we have this kind of non-host and host strain that they are totally different and how they react and interact with bacteria. So the non-host they react antagonistically, but they are able to sporulate and propagate independent of bacteria. They don't need bacteria to do that. But the host strain are addicted to bacterial presence, so they need bacteria to be there for sexual and asexual sporulation. And the cool thing is also, Andreas mentioned that we can remove bacteria, we can cure, as we say, and we can eliminate bacteria from the host strain. And when we do that, we lose sporulation. But it's also a cool feature that we can cultivate both partners independently, so we can reinfect the fungus again, and then we can recover again this population. And for the sexual exfoliation, it's the same in the case of the host strain. So if you can see here, this is a host strain to different mating types, and you can see that sexual interaction only happens with bacteria present. And we able to see those cytospores that I mentioned, those structures that are formed where sexual reproduction happens. So what we planted to do, and our goal here was to try to understand on whether there is going to be or there is exist any environmental condition that could somehow replace the function of bacteria in terms of asexual sporulation. So we tried different grow conditions, different media, and different environmental stimuli. And just to go with the most striking results that we have is that when we were growing host strains in dark, as Andreas mentioned, and it's well known in the field, we can only see sporulation when bacteria is present. These are two different host strains, but not when bacteria is absent. But when we use some light exposure, we begin to see some sporangia, and this structure contain spores. And it was a very striking result because for now, the things that we know is that bacteria is necessary for sporulation. And the dogma in the field is that bacteria is exclusively and absolutely necessary for sporulation in host strain of Rhizopus. But here, with environmental stimuli, in this case using light, we could recover the sporulation without having bacteria. And we got those spores. We look at them under the microscope, and they just look like the spores that are produced when bacteria is present. So there was no morphological differences between them. So because that the finding was really challenging what is known in the field, and this the fact that bacteria is totally required for sporulation, we need to do some parallel confirmation that the strains that we were considered to be cured and they do not contain bacteria, they actually do not contain any bacteria. So we did a PCR amplification of the bacterial 23S, and we could amplify that in host strain with bacteria, but not in host strain that we consider to be cured. Andreas already introduced the concept of rhizocin. So rhizocin is the toxin produced by the endosymbiotic bacteria and allows both partners to infect and colonize the plant. So we did HPLC and mass spectrometry, and we can detect that when bacteria is present, but not in our cure strain. And we were doing culture from the spores we were obtaining by inducing the sporulation using light. And also we compare the spores in which bacteria is present with the spores in which we are obtaining just using the light exposure. We did some confocal microscopy. We could very easily detect the spore bacteria, into the spores and germinate in the spores when bacteria was present, obviously, but we could not detect a single bacteria in the strains that we consider to be cured. So we were sure that a strain that we were using that we consider to don't have bacteria, they don't have any bacteria, and we were being able to obtain spores using just this light stimulation. So when we know that, we quantified a phenomenon, and we saw that although light did not recover all the sporulation that we see when bacteria is present, it is still enough to get some sporulation, and we can easily see that even microscopically. But also we saw that light was also inducing even more sporulation in the host strain that have bacteria. So it was having like some kind of synergistic effect on the sporulation in the host strain with bacteria. And when we realized that, we set up some transcriptomic experiments. So we compare host strain with bacteria in the dark and light exposure, but also cure host strain in the dark in light exposure, the condition that we can see sporulation. And we identified a bunch of different differential expressions. I'm not going to go into the detail for each of that, but when we kind of look at the whole response, we found that there was some common function that were commonly upregulated by both light and bacteria. So we found that activities like hydrolysis activity, transmembrane transporter, but also chitinase activity that we know is important for the formation of fungal structure like this sporangia that contain these sexual for propagation. They are both upregulated by light and by bacteria. Just a few samples of some specific genes for this is based on the knowledge that we have on the system. We have been working from the fungal system for quite a while now, but also by looking at the literature, there are some transcription factors and some genes that are well known in the fungal kingdom to be relevant for its population. For example, the white collar and velvet transcription factors, and we saw that these are not expressed in the dark condition in the host strain without bacteria, but they were clearly expressed in the case of bacteria being present, but also we were inducing with light, even with bacteria was not in there. So we conclude that light, in this case, as environmental factor, and bacteria were commonly regulated pathways that both lead to sexual sporulation and propagation in Rhizopus microspores. And one of the very cool features about being able to get spores for the cure strain just using light is that we can get sufficient amount of spores. And it was very difficult before to get that because you don't usually get those. So not that we can get spores by using light simulation. We were able to do marine infection using those spores, but and not just confronting host versus non-host strain. They have some genetically different background, but we are just testing the very same strain with and without bacteria. We can collect those spores and we can do the mass infection. And that's what we did. We did some mass infection experiments, and we were comparing spores from with bacteria and without bacteria, and we saw that the virulence potential of spores that do not contain bacteria was reduced compared to spores that contain bacteria. They were suggesting that those symbioses may also be relevant for mammalian infection. And that's something we definitely want to explore further in the lab. And that's for the first topic. And the second topic is totally unrelated, but I think it's also interesting because it can bring some cool things for the discussion. And that is the specific and totally different epigenetic landscape that this kind of fungi have, and particularly Rhizopus, but also some close related species. And I wanted to bring that here because I think raise some interesting question about how symbiosis can be relevant to understand this or to explain why this epigenetic landscape is so different in this kind of fungi. So probably you all know that iron methylation is the main DNA modification in bacteria, whereas eukaryotic organisms they use cytosine methylation as their main DNA epigenetic modification. But we know now that there are three major exceptions to this document. That is green algae, basal fungi, or early divergent fungi. We have many names for them, and this includes Rhizopus and also some ciliates. And those three group of eukaryotic organisms, they use iron methylation, the bacterial milk, as their main DNA epigenetic modification. And that's something you probably haven't heard of before in fungi because this is we did a survey and we scanned several fungal genomes and we found that this modification was only present in those basal lineages, including Rhizopus. But if not, it's not present, was not detectable in the Dicaria, and Dicaria have the most well-studied and characterized species like baker and birches and mushrooms. So iron methylation is restricted to the basal lineages only. So during my PhD, I focused on trying to understand what could be the biological implication and the role of this epigenetic modification in the genome of this kind of fungi. And when we look at the distribution, we saw that iron methylation was not randomly distributed, but it was concentrated on specific regions, and they usually appear downstream of the transcription start site on some specific genes. And because we saw that distribution, we thought that they could be somehow linked or associated with gene expression regulation. And indeed, we found that genes that are actively expressed and transcribed, we can see that peak of iron methylation just downstream of the transcription start site, but we don't see that accumulation on silent genes. And we look closer at that peak, we saw some oscillating pattern, and it was anti-correlating with nucleus occupancy. And we found that actually adenine methylation sites are located on the linker regions between nucleosomes, and it's this acting as contributing to maintaining the structural stability of those nucleosome arrays. And also, and related to also gene expression regulation, we saw that this modification is usually found on open chromatin regions, which are usually marked by S3K4 but it's absent in heterochromatin regions that are typically modified with S3K non-3-methyl in this fungi. And also, we were looking at the distribution of this modification. We saw that there were mainly two types of sites. We have the asymmetric and symmetric, and very simple way to explain this: asymmetric is when only one DNA strain is methylated. Symmetric means that both DNA strains are methylated. And this is usually happens on AT nucleotides, which is the same that happens with CpG methylation in the other eukaryotes. So basically, you have one adenine, one strand methylated, and you have the adenine, the other strand also being methylated. So when we look at this at the fungal population, we saw that we have some species in which they have predominantly asymmetric methylation, so either one strand or the other strand is methylated. But we also have some species that they predominantly have symmetric methylation, like Phacomesis here, but also this is the case for Rhizopus, which more of the sites are asymmetrically methylated. So all this is cool, but how all this is related, or why this is relevant for symbiosis, and the connection came when we were trying to identify which one were the components responsible for the deposition of this modification of fungal genomes. So at that time, we have three putative methyltransferases identified in the genome that we call MET-A, MET-B, and MET-C. And long story short, we did gene knockout, and we found that MET-B was responsible for asymmetric methylation in this fungi. But more importantly, when we did a phylogenetic reconstruction of MET-B, we found that most of the ortholog that we found were actually bacterial genes. And we realized that this thing was actually originated from an horizontal gene transfer from the symbiotic bacteria to this fungal host. So by close infection of the sequence, we realized that the glomeromycota MET-B was actually transferred by their own endosymbiont bacteria. So this might be relevant to think about further, because this endosymbiotic bacteria is also using adenine methylation as the main DNA PGN modification. So this symbiosis and this transfer is something that's definitely something that we would like to keep thinking and keep exploring why this is happening, and what evolutionary meaning have this for this kind of interaction. And that was the case for asymmetric methylation. And in the case of symmetric methylation, we also identify another genes. In this case, we identified a complex which is formed by two DNA methyltransferases. And we tried to knock out this also in Rhizopus, but in this case, we were not able to get mutants, and it was suggesting that they might be essential. And what we did was to develop a reliable expression system based on the use of zinc. So we add zinc to the media, we reduce the expression of the gene that we are regulating. And we did that with one of those methyltransferases, and we found that the mutant we were adding zinc, the mutant was not able to grow any longer, and we were reducing the expression of the methyltransferase. So there was it was supporting and giving more evidence that the fact that this complex and symmetric methylation is an essential DNA modification in Rhizopus and probably in other basal fungi. So this kind of like shows that how relevant this modification is for these organisms. And we also studied the conservation of these components, and we found that these methyltransferases are only conserved in those three groups of eukaryotic species that have adenine methylation, which is, as I mentioned before, this basal fungi, green algae, and ciliates. But it's lost in all other eukaryotic clades, in plants, in animals, in any other eukaryotic clade. So if you think about this from an evolutionary perspective, you, you can consider that these components have suffered from recurrent losses from the last common eukaryotic ancestor, and they have only been conserved in very few specific eukaryotic clades. And the reason for that is totally unknown. So we don't know why this is conserved in those particular organisms. And to answer that is what I think that those cross-kingdom ecological interactions, those symbiosis, might be important to explain why this is happening. So we consider the interaction that those eukaryotic organisms establish with other organisms, we might find an answer on why they are still maintaining this component for symmetric methylation. Because you might think, how could symbiosis explain this? So uh, the way I like to imagine and think about this is that in those symbiosis, you can think that or you can imagine that both partners are using what I like to call like a share of a common epigenetic language, in this case, methylation, and that would also would possibly allow for cross-kingdom regulatory networks in which one partner is regulating the other because both, in this case, the fungus and bacteria are using and understanding the same epigenetic language. This is, in this case, is adenylmethylation. So many open questions in the field right now, and sorry to finish with more open question and not just close conclusion as usual. But I think this kind of open question also very relevant to science to what we do. And well, with that, I would like to thanks again for the organizers for having us and being interested in on what we do. And I would like to thanks to all my collaborators and of course my research group. And I will hand it over to Andreas for his own acknowledgement and happy to take any question afterwards.
Thanks, Carlos. Yeah, thank you. A big thank you to the Euling
Sure.
Lab and our collaborators at the University of Sheffield and the University of Exeter and my funding source at the NSF through the GRFP. So big thanks. And thank you all for your attention.
Thank you so much. That was really exciting presentation. I just love those symbiosis, especially with fungi and to understand that. Um, Now I have just one quick question, and so to both of you. So for the one that living inside different fungi and cannot be cultivated, do we know anything about their genomes? How big their genomes are, those bacteria, or is there like some kind of pattern, or what they are?
Yeah, those genomes are quite small. I don't have the number off the top of my head, but I believe that the you're referring to the Glomerobacter bacteria in the Glomeromycotina, and they're like I believe they're like 1 to 2 megabases.
Megabytes.
Yeah.
I have a small question. This is more like blue skies, but is there, for each of your research, is there sort of like one discovery that you think would unlock a lot of the rest of this, or one method that's missing? I understand that like the being able to screen all of these interactions is a really useful part, but if you could invent either like one discovery or one tool, what would they be?
I'll let Carlos take that one
Well,
first.
well, I think we is I will hand it over to Thomas because I think that what these people is doing with the macroinduction and being able to manipulate the bacterial fungal interaction and being able to just put everything you want inside the fungus and manipulate that, I think it's great to understand what's the nature of this kind of relationship. So I don't know, Thomas, if you're going to comment on that, but what's your plans to keep expanding those kind of tools that you guys are developing? But I think this is something I think I don't know, Andreas, what do you think? But these kind of tools are really relevant to understand what's going on with this kind of symbiosis.
I can first start with a comment on your work because I love what you showed on the light induction of the sporulation. I think this unlocks a lot of other works because and I'm speculating now as well, but last year at the conferences, many people that do sampling in environments told that we find predominantly endosymbiotic fungi in sandy soil environments, in deserts. So and it doesn't really make sense, right? But now you show that that light predominantly triggers sporulation. So can it be that this is kind of a way of how they are selected? That light is like dominate triggering sporulation in these environments more predominantly, and so vertical transmission is favored, or selection via vertical transmission is favored in those environments. So I think this is kind of a direction that I thought now when I heard your talk and I've seen the data for the first time. So I'm kind of really intrigued by this. I'm not sure about the direction that you go with the like infection and pathogenicity of this. I rather see this really beautiful in when I think of ecology and what does it mean in the environment when you see triggers by light. Maybe I would think there.
Uh, I agree. I
Uh,
think that oh, sorry, I didn't mean to interrupt.
no, and I mean we always say that if a strain doesn't sporulate, yeah, just give it some light. No, but you show this now very clearly for the with the connection to Anderson bells, so I love it. Then for Fluidesenberg, so of course Julia Vorholt's lab in Zurich, they are working on putting different bacteria into Rhizopus, and people in the audience are here. So, but here also to say to the community, I now changed position. I'm now in London at Imperial College, and I will follow up on some of the work from Rhizopus and Ralstonia, but Fluidifem. So I don't have a Fluidifem at the moment, so I can't pick up on this kind of work directly. But Julia's lab is doing this, yeah. But I mean that I think you got this from the talks from Julia's group is that it's not a very high throughput method. So for screening, it's kind of difficult that you screen through a phylogeny of different bacteria. You kind of have to know already what you want to do a bit. You narrow down what you do, but then it's I think the most powerful tool here that we have in the community.
Yeah, I agree. It's a really amazing tool. And yeah, Carlos's finding with this sporulation, like it really does upend our understand, like what sort of like he said, like dogma within the field, and I think will be really helpful to me, at least in my work, trying to get spores for cured host strains. I think that some things that could improve this, like our ability to work in this field, would be improving our resources for manipulating the genomes. And so, Carlos has developed this CRISPR method, but I think like a whole genome mutagenesis library would be really helpful to better screen mutants within Rhizopus. Um, Improving our ability to germinate zygospores so that we could do crosses and identify like QTL marks and better link genome to phenotype. And then finally, just improving scale, as Thomas was kind of referencing. They're kind of hard to work with, and you can do scale, like there are high throughput experiments you can do, but they're fairly difficult organisms to work with at scale. So I think that would be one area for improvement.
Yeah, definitely.
Thank you, guys. I need to apologize because I will have to leave to another meeting that starts in 2, 3 minutes. So maybe at this point I will stop the recording. But we can, if anyone is interested, obviously we can have more discussion here. So thank you again for this lecture and just all this information that you provided here.